Hi, I'm Evan Palmer Young of the Bee Research Lab of the U.S. Department of Agriculture, discussing the potential for high temperatures, gut acidity, and diet chemistry to shape the distribution of parasites in insects and resistance of bees to infection. Parasites can cause dramatic changes in fitness of their hosts, driving selection for traits that provide resistance to infection. In turn, these host resistance factors become challenges for parasites that shape their fitness in different host individuals and species. Three factors that can affect parasite establishment are temperature, gut acidity, and phytochemicals. A common response to infection in animals is an elevation of body temperature. These high temperatures can ameliorate infection in mammals, reptiles, and insects. However, only a few insects possess a mammal-like ability to maintain high body temperatures for extended periods. Gut pH, shaped by both digestive enzymes and the gut microbiome, is another factor that can influence parasite establishment. pH in the insect gut can range from a highly acidic pH 2 to another insect's a strongly basic 10 or 11. Antimicrobial phytochemicals provide resistance to infection both in plants themselves and in plant-consuming animals, as evidenced by the use of various plants for medicinal purposes in humans. Plant compounds can affect infection in plant-eating wild animals too. The diversity of plant food sources consumed by different insects result in a variety of phytochemical-mediated challenges for their parasites. If these three host resistance factors are indeed drivers of the ecology and evolution of insect parasites, then we would predict that parasite tolerance of each would reflect the conditions that they encounter in their respective hosts, with heat tolerance reflecting host body temperature, pH tolerance reflecting host gut chemistry, and phytochemical tolerance reflecting the composition of host ingested plants. To test this idea, we need to compare related parasites from host niches that differ in each of these factors. The trypanosomatids of insects provide an ideal family for comparing the tolerance of related parasites that infect hosts with differing body temperatures, gut chemistry, and diet chemistry. This family of protozoans includes species that infect hosts from a variety of nutritional niches, including phloem-sucking bugs, nectar and pollen-eating bees, and blood-sucking flies. There are even a few species that infect plants themselves, as well as a handful of insect-vectored leishmaniasis species that have managed to invade warm-blooded mammals and cause leishmaniasis, a major neglected tropical disease. Now, intriguingly, the high body temperatures and acidic intracellular pH found in mammal blood cells are thought to have been two major barriers to this host shift, while phytochemical-rich plant extracts have formed the basis of traditional remedies for infection. Here at the Bee Research Lab, we're particularly interested in the parasites of honeybees, which are close relatives of the leishmania and even closer to the species that infect bumblebees. These parasites include Crithidia mellificae, discovered in Australia in the 1960s, and Lotmaria passim, named right here in the Bee Research Lab. Trypanosomatids have been found in over 80% of colonies in some national surveys and associated with colony collapse on 3 different continents. Now, a few aspects of honeybee biology make them unique among the trypanosomatids insect hosts and could challenge the establishment of parasites. First, honeybee colonies exhibit an endothermic temperature regulation that's quite rare among insects, keeping the colony at close to mammalian body temperatures. That's much hotter than what's typical of most insects, and considerably warmer also than the standard growth temperature for trypanosomatids, suggesting that bees' high body temperatures could deter parasite establishment. Second, the nectar and pollen-based diets of honeybees and fermentation of these materials by their intestinal bacteria result in a gut environment that is acidic and rich in nectar and pollen-derived phytochemicals, most notably flavonoids, which are strong inhibitors of leishmania. These acids and phytochemicals may pose additional challenges for gut parasites. Besides bees, another insect host of trypanosomatids is the mosquito. Their parasite, Crithidia fasciculata, establishes in a niche quite different from that of the bee parasites. In contrast to honeybees, mosquitoes do not maintain high colony temperatures, and their guts are basic rather than acidic. And with respect to diet chemistry, although mosquitoes do feed on nectar, they rely on blood rather than phytochemical-rich pollen for amino acids as adults. On the other hand, larvae are exposed to considerable levels of phytochemicals in the debris-filled ponds where mosquitoes breed, and the larvae sometimes browse on decaying vegetation directly. Hence, we predicted that parasites of bees would have greater tolerance of high temperature, low pH, and pollen-associated flavonoids, whereas parasites of mosquitoes would be more tolerant of high pH and compounds derived from wood decomposition. To test these predictions while also evaluating possible treatments for managed bees, we measured growth rates of the 2 honeybee parasites and 2 strains of the mosquito parasite across a range of temperature and pH, and in the presence of 26 phytochemicals, including compounds associated with pollen and with wood decomposition. Here are the responses of growth to temperature in the honeybee and the mosquito parasites in blue. Crithidia mellificae, the honeybee parasite described in the 60s, had the highest heat tolerance, and actually higher than any insect-specific trypanosomatid studied to date. Both bee parasites in red were more heat tolerant than the parasites of mosquitoes, indicating that the high temperatures of the bee colony could provide protection against non-specialist parasites. However, the emerging parasite Lotmaria was significantly less heat tolerant than was Crithidia mellificae, with a 50% reduction in growth rate over the temperature range here found in brood-rearing honeybee colonies. This suggests that turning up the heat in the colony, for example, by changing hive construction or stimulating brood rearing, could reduce infection of hives. And although speculative, the relatively low heat tolerance of this emerging parasite raises questions about whether it might have recently jumped from a different bee species with lower brood rearing temperatures. pH tolerance also varied substantially between the bee and the mosquito parasites, reflecting differences between the acidic guts of bees and the alkaline guts of mosquitoes. The honeybee parasites grew at acidity levels full 2 pH units lower than the mosquito parasites did, suggesting that the acidic guts of honeybees could limit the types of parasites that establish in their guts. The established parasite Crithidia mellificae had a remarkably broad pH range, able to grow at any pH between 3 and 10. This could explain why it's been found in a broad range of hosts, with presumably diverse gut chemistry. The emerging parasite Lotmaria, though, appears well suited to the acidic pH of the bee intestine. However, growth was essentially limited to these acidic conditions, potentially explaining why this parasite has been found in acid-gutted apis almost exclusively. Summarizing these results, the bee parasites had greater tolerance of heat and acidity characteristic of bees, whereas mosquito parasites were more tolerant of basic conditions found in the guts of their hosts. These associations between host and parasite traits suggest that both temperature and pH are potential drivers of insect-parasite associations. Now, how about phytochemicals? Of the compounds tested, 11 had quantifiable inhibitory effects on the bee and mosquito parasites. Some of these, such as thymol, have been recorded in floral nectar at concentrations up to 26 micrograms per milliliter, comparable to the concentrations needed for inhibition of the parasites tested here, and the insect vector leishmania, these horizontal lines, suggesting the potential for antiparasitic flora resources or phytochemical supplemented diets to mitigate infection in bees and other insects. The bee parasites had much greater tolerance of the flavonoid chrysin, consistent with the high flavonoid concentrations found in nectar and especially pollen and propolis. The mosquito parasite, on the other hand, was more tolerant of 2 compounds associated with wood decomposition, the benzenoid vanillin, and the phenylpropanoid, cinnamic acid. Both of these are found in larval habitats. Finally, the majority of the compounds tested, although chosen for activity against Leishmania, had weak effects on these insect-restricted parasites, even at well above the Leishmania inhibitory concentrations reported in the literature. Particularly notable for most of the flavonoids, which are present in nectar consumed by both bees and mosquitoes. However, nectar is also a favorite snack for Leishmania vectoring sandflies between blood meals. So looking at our results from the perspective of Leishmania control, susceptibility of Leishmania to these nectar-associated flavonoids suggests that floral nectar could reduce parasite loads in flies. It turns out that the reported Leishmania inhibiting concentrations of several flavonoids, shown here, are exceeded by the concentrations of the same compounds in some plants' nectars. This suggests that planting these or chemically similar plant species in Leishmania hotspots could help to reduce sandfly-mediated transmission of Leishmania between human hosts. Summarizing, the bee parasites had higher tolerance of a pollen flavonoid, corresponding to the flavonoid-rich diets and hive chemistry of honeybees, and the mosquito parasites were more tolerant of compounds associated with decaying woody debris found in larval habitats, an important stage for parasite transmission. Also, the insect-restricted parasites tested here had generally higher tolerance of nectar and pollen flavonoids than did the mammal-infecting Leishmania, suggesting trade-offs between phytochemical tolerance and infection of mammals, as well as ecological interventions that use phytochemical-rich floral resources to target parasites in nectar-feeding insect vectors of human disease. Results of our temperature and pH experiments have implications for bee management, helping to elucidate how season and brood-related changes in colony temperature could affect infection, conservation, suggesting how bees with different thermoregulatory abilities and social transmission of acid-producing gut symbionts could vary in susceptibility to infection, and ecology, using bees as models to better understand the value of endothermy as a behavioral component of immunity. Beyond the bee community, this research could also enhance our understanding of parasite adaptations that enable the jump from insects to mammals, where the pH of the intracellular habitat of Leishmania is very close to that found in the honeybee hindgut. In addition, heat tolerance is a feature that distinguishes the Leishmania strains that cause unsightly but rarely fatal skin lesions from those that infect the warmer visceral organs, causing over 90% mortality. The 4-degree difference in temperature between skin and visceral lesions is very close to the difference in heat tolerance between the mosquito parasites and Crithidia mellificae from honeybees. So, bee parasites could serve as models for development of heat tolerance in insect parasites that enables their survival in warm-blooded hosts. The results with phytochemicals also have implications for pollinator health, parasite ecology, perhaps even human health, most immediately suggesting floral resources and therapeutic treatments that could be used to manage infection in bees. In terms of parasite ecology, they provide suggestive evidence for the role of plant compounds in host diets as a driver of host parasite specificity, which could contribute to the patterns of trypanosomatid infection found in insect communities. And they underline the complementarity of research with bee trypanosomatids and insect vectored parasites of humans, with bee parasites providing resistant models for understanding parasite tolerance of clinically relevant phytochemicals, and synthesis of research on plant-based therapies for Leishmania with studies on nectar chemical ecology suggest how phytochemical-rich floral resources could provide an environmentally friendly way to reduce transmission of Leishmania by nectar-feeding sandflies. Thank you to my colleagues and sponsors, and I hope you will get in touch with questions. Thanks for your audience.